Hi there, and welcome to Mr. Fisher's Biology Podcasts. This podcast looks at the structure of DNA. So DNA is often called the blueprint of life. What that means is that simply DNA contains within the molecule instructions for making proteins within the cell. And all of the proteins that make up a living organism have instruction made in DNA. So why do we study DNA? Well, there's a lot of very good reasons to look at DNA and study DNA. First of all, DNA is important to all life on Earth. Remember, all life comes from or is believed to have come from a common ancestor, and we can begin to trace that ancestry looking at the DNA. DNA also has some medical benefits. Like cures for diseases may come out of our understanding of DNA, or even better food crops. And have a look at this article right here. You can Google this guy, Charles Artson. One of his interesting fields is looking at bioengineering foods with vaccines. So people in other countries could get immunization from diseases by just eating some of the foods that would be common in that area. So as we understand DNA, we can begin to apply DNA to medical and agricultural benefits. So first of all, let's look at chromosomes in DNA. The genes that make up a human are located on our chromosomes. And chromosomes are really just sections of DNA. So chromosomes are made of DNA. Remember that chromosomes are located here in the nucleus of a cell. And if we are, for humans, we have 46 chromosomes. So if we look at the shape of a DNA molecule, DNA is very long. It's shaped like a twisted ladder, and oftentimes we call this twisted ladder shape a double helix. One strand of DNA, if we look at DNA, is composed of alternating phosphates and deoxyribose sugars. So this right there is our phosphate, and this is that deoxyribose sugar. And the teeth are nitrogen bases. Now, one strand of DNA is made up of nucleotides. And remember, think back to our lesson on macromolecules. Nucleotides are the monomer for nucleic acids. One stranded DNA has millions of nucleotides all strung together. And there are 4 nitrogen bases that make up DNA, and those are cytosine, thymine, adenine, and guanine. And in terms of DNA studies, we tend to shorten those down into these 4 letters here. So those would account for the different nitrogen bases. So there's 2 kinds of bases that we can notice in DNA. First type of base are called pyrimidines, and pyrimidines are single ring bases. So that's a pyrimidine shape. And then purines are double ring bases. Now, thymine and cytosine, there are pyrimidines. Okay, so thymine and cytosine have the 1 ring structures, and they make up our pyrimidines. And adenine, guanine are purines, and they have 2 ring structures. And why is this important? Well, if we look at the 2-stranded DNA, remember that DNA, one strand, both strands will go together, and the bases will actually attract each other because of these hydrogen bonds. Remember that hydrogen bonds are very weak, but when we add millions and millions of them together, it makes for a very strongly held together molecule. Now, when making hydrogen bonds, this is where those purines and pyrimidines come into play. Purines will only hydrogen bond with pyrimidines, and they will only hydrogen bond with certain types. So when making hydrogen bonds, cytosine will only always pair up with guanine, and adenine will always pair up with thymine. Now, a fellow by the name of Chargaff came up with Chargaff's rule. And Chargaff's rule is simply just that. When pairing in DNA, adenine will always pair with thymine, and cytosine and guanine will always pair together. In this way, a purine is always pairing with a pyrimidine. And that concludes our podcast on DNA structure. and I hope that was helpful.